A cognitive schema is often defined as organized pattern of thought or behavior that organizes categories of information and the relationship among them. In short, a schema is a clearly delineated network of nerve cells containing the information of a defined behavior, cognition, or emotion. An example would be how to ride a bicycle, knowledge about an object like a football, or the emotion that arises from fear of snakes. Such schemas are extremely important because they lay down guidelines for how we perceive the world around us. They are essential to our perception. Similarly, a schema's ability to adapt to new knowledge is important. A disturbed personality will therefore, due to their schema, understand the world in a different and more rigid way than a person who is normal and who have less vulnerability and greater flexibility in their schemas. Such schema structures can contain anything that could possibly be stored in nerve cells. Our language comprehension occurs by activation of schemas, our visual understanding of the same. Our ear interprets sound using schema structures, and we produce language based on separate schemas. Different nerve schemas cooperate and control our behavior. Cognitive schema is the organization of knowledge about a particular concept. The concept of cognitive schema was introduced by Jean Piaget in 1926. He said children were born with a set of basic mental structures that were the basis for all learning. These were gradually filled up and new ones were added. A cognitive schema is thus a nerve structure built on biology in the form of nerve cells, chemistry in the form of neurotransmitter and proteins, and physics in the form of electricity. It constitutes an organized pattern of nerve cells which have a content that defines, for example, memory of a theme, emotion, situation, or an object. A type of schema we use in everyday life is how to get from A to B. If you take a cab and say you're going to the airport, the taxi driver pulls up a mental map of where he needs to drive to get from the base to the airport. We can say he has a cognitive schema or map of where to drive. The neuron is the building block of a cognitive schema. This is a typical nerve cell. When multiple nerve cells are connected together in large structures, we get a network called a schema. This may include cognitive and memory information or may contain emotional memory. This is a small segment of a cognitive schema. It contains a large number of nerve cells, often millions, which are connected in parallel and in series, and if activated, give us a consciousness, emotion, or behavior. It can be about a particular behavior, such as how we bicycle. It may be knowledge about a person, a situation, an object. It can be how we understand a particular behavior or observation. There may be directions we mentioned above in the taxi example. When we see an object, the brain will try to understand what we see by comparing the sensory input with our knowledge that we have present, our present cognitive schemas. The brain searches existing cognitive schemas to see if they are activated by sensory input. If they do, it is experienced as recognition. Have we seen or experienced this before? We recognize the sensory input. We understand what we see. When we hear a melody, the brain will likewise search in memory, for example, the cognitive schema, and try to recognize the melody. We try to find the schema for the current song. If we recognize the melody, we will know how the melody goes before it's played. It has triggered our cognitive schema for this melody. It was recognized. When we see a movie we recognize, we know how it ends because the cognitive schema for this movie is activated. When we turn on the radio and hear a sound, we will immediately try to make it understandable. Is it a call, a song, or what is it? Our cognitive schema helps us in this process. So it's all about finding association to sensory input. It also means that we will be conscious of schemas with a content similar to sensory input. The same happens if we experience a recognized behavior, then we will activate the schema we associate with such behavior. And of course, schemas for similar behavior is also activated. It is important to be aware that a cognitive schema may activate other schemas. For example, we see an object we recognize, while emotions associated with this object is activated. The other way around, emotions will make us start thinking of a cognitive schema of an event associated with this emotion or object associated with this. A song can activate the memory of a dance or the memory of where we were when we first heard this song. Sense impressions can also activate the form of an experienced trauma, which in turn activates the emotions associated with this trauma. The smell, the sights, surroundings and sounds that were present during a rape would later be associated with such a trauma. A bed in a white room can activate emotions about abuse one has experienced in rooms that give the same associations. If sensory impressions similar to memory of trauma activates the emotions associated with the trauma, one can get aversions, which in itself is harmless, but if the association is to a real trauma, negative emotions arise. This is cognitive schemas in practice. Also, separate experiences related to an object will be activated. If we see something we have no memory of, the brain will activate a new cognitive schema for this sensory input. Young children develop such schemas in tremendous speed and constantly corrects existing schemas throughout the process of accommodation. Two concepts are important in the context of schemas. Assimilation means that we use existing schema to understand the sensations. The world is experienced according with our memory of the world. This is about adapting the world to oneself. A child goes to bed because it gets dark. This is transmitted to the understanding of the sun. It's night because the sun goes to bed in the evening. The second term, accommodation, a new cognitive schema is produced or existing schemas corrected. It is about adapting oneself to the world. The child learns that the earth goes around the sun and around its own axis. Therefore it becomes dark. Schema for why it gets dark must be corrected. These processes run in parallel. People with severe personality disorder, such as narcissists and psychopaths, have greatly reduced ability to correct their cognitive schemas. For narcissists, particularly important in the schema attaching to their self-image. This is often caused by the schemas or strategies they have developed. Not only does the memory contain what actually happened, but it also a defense against an intolerable pain after an early experienced trauma. Associations of early trauma affects their behavior. We can say that they have cognitive and emotional schemas they fear activated because they feel so anxious and painful. Schema structures activates negative affect. Meanwhile, other schemas are poorly developed. For example, those who those who are connected to the ability of mentalizing. To better understand the complexity of this, we shall see how these structures are built and working. Different types of schemas, cognitive or emotional, consists of a certain number of nerve cells connected together. This is a typical nerve cell connected with other neurons to form a network, a schema. In the brain there are an estimated over 100 billion such neurons, and each of them can be connected to 10,000 other neurons. It constitutes a formidable network of storage space for memory in cognitive schemas. Our memory is stored somewhere in the nerve cell. There are many different types of nerve cells and they have different functions. Let's look at some of the most important parts of the neurons. In the nerve cell's nucleus we find the chromosomes, DNA molecule called up on many small protein spheres. This is our genetic material, our DNA. It provides a roadmap or blueprint for all the body's building blocks and functions. DNA is the overall pattern of protein structure and composition, and proteins are the body's building blocks. They are composed of different amino acids in complex chains. The cell nucleus is surrounded by the cell body, which also contains a number of other components and functions required for cell growth and maintenance. From the cell body, a series of tentacles called dendrites are connected. Each of these dendrites can have multiple branches, and on those we find the synapses, connection points to other nerve cells. Their function is as receiving points for the other nerve cells. There can be several thousand such receiving points in a nerve cell. Based in the nerve body is also a long outgrowth called axon. It terminates in a branch of contact points to other neurons called axon terminals. It is throughout this that the neuron sends an electric signal that activates other neurons. Axon hillock is the starting point for the nerve cell's axon and determines when a cell fires, when an electrical signal moves throughout the axon. This is where all signals from dendrites gather before an electrical charge moves down the axon. Both inhibitory synaptic potentials and excitatory synaptic signals are collected here. The sum of these determines whether a charge moves down the axon, passing the triggering threshold, thus increasing by 15 millivolt from minus 70 millivolt to about minus 55 millivolt, an action potential is triggered. Axons are surrounded by an insulating layer of fat cells that makes the electrical signal travel faster throughout the axon. This layer of fat cell is called myelin. Myelin is divided into separate parts so that axon surface periodically comes in contact with the external environment. This contact with the surrounding ions are essential for nerve cells' ability to send signals to other nerve cells. MS or multiple sclerosis is a disease that occurs when the myelin sheet is broken down or disappears. Then the signal goes more slowly, resulting in uncontrolled movements. We can compare a nerve cell with an iPhone with earphones. iPhone is the cell body. In it there is connected a cable that leads an electrical signal to the earphones that converts this signal to the music we hear. Axon represents here the electrical cord. Between each myelinated part of the axon there is a small opening called the node of Ranvier. It is in these nodes that the activity takes place, which ensures that the nerve cell's electrical signal quickly moves down the axon. Where this myelin sheet is damaged, nerve cell signals are slower. At the end of the axon we find the axon terminals, that is, connection points to other neurons or muscles. Here the electrical signal that goes through the axon ends. Here is another representation of the neuron with all its inner components. As we can see, it is far more complex than what I have described here. Cognitive schema is therefore a highly complex structure that can be affected in many ways. But let's look at what is important for our understanding of what a schema is. The connection point between two neurons occurs where the axon terminal from one nerve cell, presynaptic neuron, meets the dendrites of the other neuron, postsynaptic neuron. The two nerve cells are not touching each other physically. Between them is a small gap called the synaptic gap. This is a very important gap because from what we know today, it is in this area our memory is stored. New connections between cells, synapses, are formed when the brain is active. This means that new axon terminals are developed as new knowledge is stored. Likewise, are new protein channels produced when a new memory is stored. Let's take a closer look at this. The dynamics of this gap is extremely complicated, and I will here only describe some part of it. It is in this gap one mainly can affect neurons. The concentration of so-called neurotransmitters, signal substances, determines whether a signal continues in the postsynaptic neuron. Some chemical agents allows the concentration of neurotransmitters to stay high longer. Other substances reduce the concentration of neurotransmitters. In other words, the memory can be affected. Essentially, the following happens. Throughout the axon comes an electrical signal. When this ends in the axon terminals, it causes small vesicles with neurotransmitters released into the synaptic cleft between the presynaptic axon terminal and the postsynaptic dendrites in the receiver neuron. The released neurotransmitters attaches itself either on a receptor or a effector protein in the cell wall of dendrites. The neurotransmitter's function is to affect proteins that sits in the membrane surface of the dendrites so that they open up the ion channels that let the ions in to the postsynaptic neuron. Ions are charged molecules and particles. When sufficient ions have entered the recipient neuron, they affect the negative internal voltage so that the receiver neuron is activated and sends an electrical charge on throughout its own axon to the next neuron or muscle. Let's look at what happens in the cell wall where these proteins are located. In the plasma membrane or cell wall of the receiver neuron, the dendrites, we find two types of proteins, a receptor protein and an effector protein. When neurotransmitters come from the presynaptic nerve cell's axon terminal, one of the two things happen depending on the individual protein's functioning. Either the neurotransmitter attaches to the receptor protein, which initiate an often very complicated biochemical process within the recipient cell. This process then leads to the formation of an effector protein that adheres to the plasma wall and opens a passageway allowing ions to flow into the recipient cell. In other words, this process enables DNA to produce proteins. Alternatively, the neurotransmitter is attached to an effector protein so that it opens so that ions can wander in and affect electrical voltage in the receiving neuron. In this case, we see that the protein is already in the cell membrane. In the first case, the protein was formed. If the impact is sufficiently large, it activates an action potential so that this neuron sends an electrical signal to the next neuron or muscle. Then there are some neurotransmitters, such as GABA, letting in negative chlorine minus ions, thereby inhibiting activation of axon potential in the postsynaptic neuron. They increase the internal negative voltage relative to positive ion, which reduce the negative voltage. The proteins which sits in the plasma membrane are only susceptible to a particular type of ions. There is thus a protein for chloride minus ions, another for magnesium plus ions, and another for potassium plus ions, etc. As far as we know today, it is these proteins in the plasma wall that are carriers of our memory. Activation of these proteins give us consciousness. Activation of a number of such proteins in different neurons is experienced as an awareness of a memory. What process that gives us this awareness of a memory is not clear. Before we see how these proteins are produced, we will look briefly at what gets the electrical charge to move down the axon and into the axon terminal. This is a longitudinal section of an axon. There are 4 different types of ion channels. Passive channels without energy allows ions to pass in and out of the neuron. For example, narrow sodium channels. Sodium-potassium pump pumps 3 sodium out and 2 potassium in. This requires energy to function, namely the action potential. Voltage control channels that open and close based on voltage. Volt passes the membrane and then open these channels themselves. Ligand, which is small molecules, ion channels, which open when a ligand connects to the ion. An ionic charge entering through protein channels in the axon may be either positively or negatively charged. We have therefore ions that are activating and ions that are inhibitory ions. The number of such protein channels in the axon is between 1000 and 2000 per micrometers. 1 micron is 1,000th of a millimeter. So 1 millimeter is 1000 micrometers. That is 1 to 2 million protein channels per millimeter. Outside the axon has excess of positive ions, here indicated by sodium ions in red. The axon has a negative charge related to the outside. This is approximately minus 70 millivolt. In the axon wall there are a number of proteins that act as channels for the different ions. In resting state, most of these protein channels are closed. These are channels that are given at a given time opens, allowing ions to wander in and out of the axon. When the neuron throughout its dendrites have received sufficient ion charges so that resting potential of minus 70 millivolt increases to about minus 55 millivolt, the neuron will trigger an action potential that makes this electrical charge move along the axon. When an action potential is triggered, the electrical charge moves from one node of Ranvier to another through the following process. Sodium channels in the axon membrane open, allowing sodium ion flow into the axon. It gives us a state of depolarization, where internal charge rise to about plus 40 millivolt. This goes very fast, and within 1,000th of a second we have a condition of the action potential. The way this charge moves is that protein channels close at a certain time while they open on the next point of such a node of Ranvier. In the node the charge has passed, potassium ions are pumped out so that the polarization is restored. During a recovery period, sodium is pumped out while potassium ions are pumped in. 3 sodium plus ions are pumped out for every 2 potassium plus ions pumped in. In this way, the electrical charge moves through the axon. In humans, there is a limit on the number of charges that can move through the axon. It is approximately 300 impulses per second. After this action potential has passed, the axon recovers the axon resting potential. As one understands, these signals goes very quickly. Here we see graphically this action potential. First, the inner charge rise to a threshold value of approximately minus 55 millivolt as a result of the overall ion impact. Then an action potential is triggered. Then we have a repolarization before we get a brief moment of retrieval where no signal can go. Then the neuron is back to a resting state. If the internal charge do not reach minus 55 millivolt, no action potential are triggered. When this electrical charge that has moved down the axon reaches the axon terminal, it leads to the release of neurotransmitters into the synaptic gap. They are stored in small vesicles. The neurotransmitter works here as a key that opens the protein channel throughout which charged particles, ions, can pass in and influence receiver neuron to fire the message. Neurotransmitters can also function by closing proteins. The electrical signal that has come down the axon ends in the terminal and continues through chemical neurotransmitter released into the synaptic cleft. The neurotransmitter does not move into the receiver neuron, but acts like a key that opens and closes ions in the form of proteins. Some of these channels require several different neurotransmitters binding to them before they are activated. Selective serotonin reuptake inhibitors, so-called SSRIs, popularly called happy pills, are used, for example, against depression. Their function is to influence the concentration of neurotransmitters in the synaptic gap by preventing the reuptake of the presynaptic neuron. This helps those substances so concentration of serotonin in the synapse remain higher longer. Here are some examples of these neurotransmitter molecules. Lack of serotonin can lead to depression and anxiety. Dopamine is essential for movement and motivation, but also for the memory system. GABA acts by opening chloride ion channels in the postsynaptic neuron. Chlorine ions, negatively charged, will flow in and make the inside of cell membrane more negative. This means that the threshold for activation will be higher. GABA helps therefore to prevent triggering the action potential. Norepinephrine is important for the reward system in the brain as well as in decision making. The formation of the proteins that sits in the membrane occurs when the DNA molecule of chromosome is subjected to a signal from the environment. DNA is wrapped around histones, which largely determines when a DNA molecule must be switched on and off, what is called epigenetic. Here is another picture of histones. Here I'll simply show the process of protein formation I described above. An external signal from the environment affects the protein that is like a protection around the DNA, so that the DNA is exposed. After that comes the process of copying the DNA into mRNA. This copy of the DNA contains instructions on how that particular protein that it is a prescription for is formed. mRNA contains information about which amino acid forming the required protein. As we see, the environment activates the production of the protein, which then again do a certain kind of work, for example in the form of ion channel. The environmental impact therefore decide how the gene expressions appears, the so-called phenotype. This protein, shown here by beta-actin, attaches itself to the plasma wall as we saw earlier. An example include the number of people with obesity in the world, which have doubled in the last few years. We also know that it takes thousands of years to change the genetic code, so the increase is not only determined by genetic factors. The main cause of obesity epidemic is another: diet and reduced physical activity. The environmental impact of the genes we see in many different settings. Obesity is typical. All genes are like a recipe booklet. Which recipes that actually end up on the table represents the gene expression. The environment around us is involved in determining the activity of some of our genes. If you eat bacon for breakfast, the expression of genes are different than if you're eating crackers. The genes can actually be turned on and off like a light switch, depending on the need at any time, so-called epigenetic. These proteins produced from mRNA consist of long chain of amino acids, here exemplified by insulin and beta-actin. The interesting part of the beta-actin protein is the finding that this is formed in connection with the storage of memory. One can therefore say that outside influence in the form of new knowledge affects our DNA to produce proteins that are directly related to the specific knowledge. This protein is located in the membrane wall. Research shows that repeated stimulation of nerve cells strengthens the synaptic coupling through the production of beta-actin proteins attached to the cell wall. Memory is thought to be encoded as stable, long-lasting synaptic connections are formed between neurons that are connected to each other. One could say memory is stored by the formation of proteins in the nerve cell membrane. Retrieval of memory happens when these proteins are activated. Each protein contains, of course, not a complete memory of an object. To recreate a complete memory of an object or event, thousands, maybe millions of such proteins will have to be activated in a variety of synapses. Such activation causes consciousness of the object, or situation, or emotion. Research has shown that some neurons contain only portion of a memory of an object. The more complicated the object, the larger the nerve network, cognitive schema, is necessary to keep such a memory. One can compare this with Lego pieces, each of which poses no concrete object, but put together in a certain way, in a schema, make up an image that are recognizable. The combination of a certain number of neurons in a schema forms a memory image. Interesting in this context is the research that shows that behavior parents are exposed to can be found in the genetic makeup of the offspring. In other words, parents' trauma being inherited by the children. Of course, it's not the trauma per se, but the vulnerability for a particular behavior that is inherited. Dias et al. 2013 found that the offspring of mice that through classic conditioning had paired a traumatic experience with the smell of acetopenone had increased neuroanatomical presence of receptors for this substance. Environmental information was thus inherited through DNA to offspring. This opened up interesting perspectives in terms of memory and cognitive schemas. DNA consists, as we know, of different combination of 4 components: adenine, thymine, guanine and cytosine. DNA methylicing is a biochemical process in which a methyl group CH3 is attached to cytosine or adenine in DNA. It makes it harder to make proteins from the gene when this is connected to the gene. A hypomethylized gene has reduced the number of methyl groups attached to it and can be considered to possess a greater potential for expression compared to a hypermethylated gene. In sperm from mice exposed to classic conditioning pairing between trauma and acetopenone, one found hypomethylated genes associated with reduced methyl groups. The association with traumatization had created genes with fewer methyl groups attached, and those genes that more easily produced receptors protein OLFR151. A reduction in the number of methyl group attached to the DNA means that it is easier to make this protein from the gene. It takes less environment activation. Through association learning, classic conditioning was the smell of acetopenone paired with an electric shock when they appeared at the same time. The fear of electric shock are now produced by the smell of acetopenone alone. This led to increased responsiveness of acetopenone. It is shown earlier in the increased production of acetopenone receptor proteins. Smell receptors genes like OLFR151 is affected, so the number of methyl group in this part of the gene is reduced. The offspring of these F0 parents showed increased activation. They have thus several acetopenone receptor proteins, and their DNA had, due to lack of methyl group, lower threshold to be produced. F2 generation were given the same conditional fear response schema in the brain in contact with acetopenone. This effectively means that the genetic basis for development directions can be inherited by offspring through hypomethylized DNA. It is important to remember that the gene has not changed, but the number of methyl groups attached to the gene as a result of trauma, the electric shock, is reduced. It lowers therefore the threshold for production of proteins from these genes. The mouse was more susceptible to acetopenone, and with the smell of acetopenone follows the associated fear. Research has thus shown that chronic stress, particularly in early childhood, can change the so-called methylation process and lead to changes in the DNA chemistry. This may turn off genes and prevent the brain from a proper management of stress. Researchers have drawn a link between this chemical dysfunction and the development of chronic health problems such as depression, obesity, diabetes, and the like. This means that parents exposed to trauma may transfer an increased vulnerability to their children, through genes that have low thresholds for the production of these proteins. The cognitive schema a child develops can be affected by the stress parents experienced as young. and that child then inherits the DNA and social heritage. According to this research, parental trauma inherited by the children then develops into distorted cognitive schemas in the children. Let's use the example of Wenche Behring Breivik, the mother of Anders Behring Breivik, killing 77 people in Norway in 2012. Her mother suffered from severe personality disorders in the form of schizophrenic. This has affected Wenche's growing up, both through inheritance and the environmental impact of her mother. She was obviously traumatized. On the basis of her inheritance from her mother and her traumatic childhood with her deranged mother in Kragerø, one can assume her DNA was affected and she developed her deviant cognitive schemas. We know she had a borderline disorder. She was repeatedly admitted to psychiatric hospitals. There is every reason to believe that Anders' deviant behavior and subsequent diagnosis, narcissistic personality disorder, may have its origins in both heritage and his mother's influence. As we saw, the mice inherited increased sensitivity to acetylphenone, so is other fear impulses inherited, such as fear of loss, threats against its own self, etc. Psychologists who examined Anders when he was small described such problems very early. There is every reason to believe that Anders may have inherited DNA disruption in methylicing of genes, which through the environmental impact of his mother has led to development of the personality we saw unfold on Utøya and subsequently in the courtroom. The cognitive schema Anders Breivik developed must therefore assume that builds on the heritage and his mother's influence, the environment. So why is this so important to understand? It is important because this shows how the child's developmental environment helps to determine how the child's brain develops. The child's innate brain structures, the schema, like temper, anxiety and fear, are affected by DNA plus the environment the child grows up in. This is called epigenetics, that genes can be turned on and off where the phenotype change but where the genotype is the same. Trauma in past generations have consequences for children in later generations. How did this boy's upbringing and development, influenced by his mother, end up to be this grandiose mass murder? All cells in a multicellular organism contains the same genetic material, yet there are major differences in how the different cells look like and what their role have. The skin cell, nerve cells and muscle cells in an individual contain the same genes. The external impact on the gene is crucial to the outward behavior and appearance, the phenotype. Innate structures or schemas is filled with content determined by the environment's impact. Later developed structures holds the schema that is a result of the influence the child is exposed to. Caregiver's influence contributes to what genes that is activated. The genetically determined development progression is influenced by the environment. With good care, the child focuses on growth. Under poor conditions, the child focuses on defense. It naturally characterizes the schema's development. The child develops strategies to meet their needs during childhood, and these are a result of the influence the child is exposed to, and thus activates genes necessary for the production of the proteins that can best meet the child's need and strategies for survival. Again, the environment affects the formation of the child's schema. It is such knowledge that is the basis for the discussion when a child should start in kindergarten. Is it okay for a 9-month-old child to be in kindergarten? What does the kindergarten's quality mean to such influence? What does the increased stress on the child's neural development means? The genes alone do not determine the child's behavior. It is the environment's effects on genes that do this. A child may have genes for many developmental directions, but the direction is determined largely by environmental influences. This is called epigenetics. It turns a gene on and off. You may have genes for many different development paths, but if these genes are not activated, they will not produce the proteins necessary for such a development direction. For example, a person may have genes for schizophrenia, but if these genes are not activated, the person does not develop schizophrenia. There may be genes for aggression, but without the right exposure, no aggressive development. The same applies to a variety of behavioral traits. We all have a vulnerability to humiliation, but if we do not get offended, then we don't develop specific strategies or schemas to protect against this. Strategies that often go on to form artificial, grandiose self-perceptions. Thus, personality becomes an expression of the individual strategies to enable to survive and develop in the environment. The same applies to depression, anxiety, and several other conditions. Narcissistic grandiosity is no exception. In the Middle East, many generations have been oppressed by dictatorial governance. This has led to a development where the individual develops strategies and forms or schemas to survive. Aggression, extremely large violability, low thresholds to offend others, oppression, etc. All these are schemas to deal with life's challenges and stress. This is clearly expressed as they do not have any barrier to violate others' God image, but when their own gods are violated, then they explode in rage. This inconsistent behavior is typical for injured people and is based on the schemas they have developed through their development. It can certainly be many reasons why a child has developed fear of Santa Claus, but the children's behavior shows a schema, a strategy in such settings. While a young child does not respond, an older child shows fear. It shows that the schema for this fear has developed over time. When the child exhibits emotions to the adult's response to this will determine the child's developmental brain structure and cognitive schema. If the adult is able to take up and be a container for the child's emotions, and so soothe the child will feel secure. This will create a cognitive schema for future emotions. The child knows that it can seek understanding from the adults in situations that produce different emotions. If, however, the adults react with uncertainty, frustration, and aggression, the child will develop a cognitive schema that indicates that it is not met with understanding from the adult regarding the experienced insecurity and fear. Such cognitive schema will develop another nerve structure than if the child felt safe. This has consequences for future experiences and the perception of these. This pattern of response will shape the strategy and cognitive schema the child will use in future similar situations. This in turn influences whether the child's attachment style are secure, anxious, avoidant, or disorganized. And attachment styles are schemas. Children whose cognitive schema creates negative strategies will have developmental disorders. They will learn to pull away from situations that they associate with their trauma. Past children will pull away from the normal parent to avoid being exposed to the disturbed parent's aggression. Such cognitive schemas will continue to the rest of their life and create difficulties in relationship and work. A child growing up with a narcissistic parent will not develop a schema for a healthy self, but a schema that creates a false self. The worse the condition, the more primitive their schema, and the more primitive the person's psychological defenses. The more rigid schema and strategies becomes. In adulthood, it is difficult to change them. Also in other cells than neurons, this process occurs. A typical example of how DNA forming proteins after having been influenced by an external signal is insulin metabolism. In the cell wall we find the insulin receptor proteins. When insulin connected on such receptor protein, it triggers a process within the cells. The process ends with a glucose transport protein being formed. Pattern recipient lies in the DNA. This glucose transport molecule sits in the cell membrane and allows the glucose to enter the cell. The cell's energy needs allows a signal activating the part of the DNA which produces a protein that can open the cell so energy in the form of glucose enter the cells. Simply explained, in diabetes 1, not enough insulin is produced by adrenal cortex. The insulin-producing beta cells are damaged by, for example, chronic high levels of stress hormones. This stops the production of glucose transport proteins. No glucose enters the cell. The medication is to supply the body with artificial insulin. With diabetes 2, insulin receptors are damaged, so they do not initiate any process to form glucose transport proteins. Glucose is prevented from entering the cell. For both types, the cell fails to take up glucose from the blood. Therefore, the cell gets no energy to supply its processes and damage occurs quickly. Diabetes 1 develops mainly in younger people and, as Schema et al. 2002 until 2006 shows, has one cause in chronic stress over a long time. Constant elevated stress levels is a result of schemas and strategies to survive in a negative environment. This leads to chronic high levels of stress hormones. Such children are walking on eggshells regarding their caregivers, and their cognitive schemas develops accordingly. Here is a typical setting where children live under such chronic stress. Narcissistic dynamics. Mother hates her father for abuse in her childhood. He raped her, traumatic. She hates herself. After divorce, this hate against her own father is projected at her ex-husband. He violated her by leaving her in the same way her father insulted her through abuse. The common denominator is the feeling of humiliation. The cognitive schema for humiliation is activated. She hates the feeling and anybody who brings up that feeling in her. The boundaries of the father and her ex-husband is mixed. Boundary problems arise, visitation is sabotaged. Children become pieces to frame the father, who really become a substitute for the mother's own father. Problem with self-limits for these people are well known. The children are used against the father. Thus, we have a state of chronic stress. Past dynamics are not rare in such complex conflicts. This dynamic is typical of narcissistic personality disorder. Children who live with such people are damaged if they do not have a normal adult who can help them meet their needs. Their cognitive schema develops in order to survive under such conditions of narcissistic exploitation. Cognitive therapy is based on changing cognitive schemas. Some of these schemas may change over time, others remain unchanged. Some schemas are conscious, others unconscious. Such a schema is often an automated response pattern of an external influence. It's important to understand what a cognitive schema is, because they often form the basis for behavior and reaction to behavior. They form the basis for how we perceive the world, our perception. Actually, our schemas form our personality. Primitive, rigid schemas will trigger hostility from the environment, and we have a vicious circle that reinforces these maladaptive schemas. Observations that gives association to a cognitive schema can lead us to believe that observation is similar to the contents of such a schema. We can easily assimilate, which means we can easily misinterpret an observation. Dynamics around the psychological defense proactive identification has shown that we can easily misunderstand an observation because we so easily grab the automated cognitive schemas. It's all about psychological defenses against schemas of anxiety and fear. Knowledge of the cognitive schemas is important for understanding human development and response to sensory input in the environment. Understanding this helps us understand why people with personality disorders like narcissists and psychopaths have problems changing their behavior. Their cognitive and emotional schemas are so inflexible and containing great anxiety and fear from early traumas and chronic stress. Schemas of great shame is the narcissist's Achilles heel. I hope this description has been helpful for you in understanding more about what a cognitive schema is. Thank you.